Yeah,
directly.
sure. Thank you so much.
Yeah, let
Yeah.
me paste my email in your chat. Okay, I just sent you my email
Yeah.
address.
Yeah, I got it. Yeah. Thank you so much.
Hi, Dr. Shafi, can you hear me?
Yes, I can hear you. Can you?
So can we start our today's lecture?
Yes, we can start. Thank
Okay,
you. I mean,
so hello everyone. So welcome again. So today we are going to start our session. So the focus of our today's session is RNA-seq analysis. So we will go through first how actually RNA-seq analysis work, what approach we will follow, and then we will move to the coding session. So as you know, why we extract RNA. So if we want to see under specific conditions or inside specific tissues, what type of genes are expressing. So to understand that, we use just a simple technique of RNA-seq analysis. So we extract RNA and then we have multiple platform for sequencing. So once we got our sequence data, so what will be how it works? So this is RNA molecule. So how they sequence our data? So for example, this is RNA. So what they do, they will prepare library, and the technique we have nowadays, most common, is short read sequencing. So first they will break this into small pieces. So why they are breaking it into small pieces? Because of the machine limitations. So short read sequence has limitation that it can sequence 100 to 200 nucleotide base pairs fragments, not greater than that. So the emerging technologies that most will be dominate in couple of years is long read sequencing, where you can sequence a long fragment of RNA. So if you have ever heard about uh, RNA integrity number. So this RNA integrity number tells us, so what is the size of approximate fragment in our RNA? Is the RNA is intact or not? If the RNA is degraded, it means it has already broken down into very small pieces. So, but when we are sending our RNA samples for sequencing, we keep in mind that our RIN score should be equal or greater than 7. So if the RN is intact, then we then we then we send it for sequencing. So no, coming back. So what the short read sequencer do? It approach to, we will just first break down all our RNA into small fragments. This process is called library preparation. So when the small fragments, then they sequence these small fragments. So when we get our raw data, it should be in the form of small, really small, 100 to 150 or 200 base pair uh, base pair fragments. So this raw data will come in the form of FASTQ format. So this FASTQ format data, then we will process it. So what approach we use? Yeah. Okay. So here is the overview of RNA-seq analysis approach. How we gonna do it? So the first step is, as we have already learned, that the reads we will get, these will be really small reads. Each samples, each samples will contains millions of reads. Sometimes we get billions of reads. So these reads will be really short, 100 to 150 nucleotides. So first thing, what we're gonna do, we will remove the small sequences. Like if in our samples, the sequencing reads that we got, these are in millions. So first we're gonna remove shorter reads. That can be 100 base pair, 120 base pair, or 30 base pair. So mostly we set a quality score. Okay, remove sequences that are less than 50 bases. So we will remove. So what we use, there are many packages that we can use to remove the short sequences. So for this webinar or lecture, we will use StreamGlair. That's a Linux package that we are going to use. So other thing, when we are doing sequencing, short read sequencing, attach adapters to the small fragments. So we also need to remove these adapters, and the stream glare, it can automatically remove the adapter sequencing. So next step is alignment. So if I gonna share again the whiteboard. Okay, for example, these are the short reads in our sample. So we have removed the adapter, we have removed filtered the short reads. So next, what we are going to do, as you know that these are really short fragments, 100 to 150 base pairs fragments. So what we need to do, how we will make sure that, okay, we are going what the final reads we are getting, it are correct. So what we will align it to our reference genome. So for example, we are doing RNA-seq analysis of mango tree. We need a reference genome. It should be a reference genome of mango. So we will align these all small pieces with this reference genome. So once we are going to align so no these small pieces has been aligned with the reference genome. So this is called alignment. Okay. Once we have done alignment, we got millions of reads. So all reads will be aligned. So these are the reads that are aligned. So this one is reference genome. For example, this one here, one at the one part of this reference genome, 50 sequences has been aligned. And the other part here, 100 sequences has been aligned. Okay, so this is a sample one. For example, this is the sample one. Now we have similarly sample two. In sample two, here 70 reads has been aligned, and here 30 reads has been aligned. So for example, this one is the gene one, and this is gene two. So know what will be our expression in sample one. Gene one has been expressed expression is 100 and sample in sample 2 gene 1's expression is 70. Similarly, this one is gene 2. For example, at gene 2 in sample 2 the gene 2 has 50 reads and in 30 reads, and in sample 1 gene 2 has 50 reads. So what will be the expression score of gene 2 in sample 1 50, and in gene 2 the expression score will be 30. So after alignment, we need to find out, okay, how many reads has aligned to gene 1, gene 2, or gene 3. So this is the simple approach. Now we will learn how we can do coding to do that. Okay, so the alignment for alignment, the tools we have star, his two, and bowtie two. So these are the alignment tools. Okay, after alignment, no, we do quality check. Okay, how many percentage of reads has been, what is the alignment score? So we can do alignment score using SAM tools or PyCard. These are the packages that we can do. So alignment scores means, so if, for example, a genome is 30 megabase, how many percentage of reads has been aligned? Either the reads has covered the whole genome, or it has been partially attached. So the quality control will let us know. So next step is quantify. So quantification, how many reads has aligned to a specific gene. For quantification, we use the package feature count or HTC. So after we got the count number, so then we will adjust the reads depending on our genome size. So method we use is TPM or RPKM or FPKM. Yes, the percentage means sequencing depth. Okay, at the end, we will do differential expression. So differential expression, we use package D, SIG2, or EdgeR. So these are the packages we use for differential expression. And the next step is functional enrichment analysis. So there are two approaches to do that. Functional enrichment analysis can be done using R packages, and there are other web interfaces that we can use like Shiny Go. we can use that and do our functional enrichment analysis. So this was the overall approach we are going to use today. So now we will move toward the coding part.
Hello, can you hear me? I
Yeah,
have a question.
sure.
You talked about the reference genome for analysis,
Yeah,
but if we are analyzing something new, it's you talked about the RNA sequencing, but in case of DNA, but in some viruses we have RNA genome. If it's the new species, what would be the reference genome?
that's a really great question. So we have also in agenda de novo assembly. So we do de novo
Okay,
assembly.
okay. Thank you.
Yeah. So I when I started working, so the project I was doing, I sequenced my samples, but we don't have any reference genome at that time. So I first started with de novo assembly directly. So we will learn that. So here's the pipeline we are going to use for this RNA-seq analysis. So you can see the first thing what we are going to do is check the quality. So for quality, we will use the package called FastQC, and then running FastQC on all our samples. Then we will run the another package that's called MultiQC. So FastQC, it will analyze all our files, and then for generating report, we will use MultiQC. So MultiQC, it will generate a report, then analyzing report. Then we will decide, okay, what approach we gonna follow for trimming and filtering our reads. So now I'm going to open my PowerShell so we can go through the coding part. Okay, first thing first. So how we gonna install multi QC? Simple. We have already set up our environment. So it's really simple. No installing something with Conda. So you can write Conda install. So you can see because I have already installed, so I don't need to install it again. So I'm stopping here. So this is how you can install FastQ, and then
Please repeat the procedure for installation of FastQ.
okay. I just pasted in the chat. Okay. So how we are going to do it?
Excuse me.
Yes.
Yeah, can I share my screen and show you? Where's the error? I can't do it. I don't know.
Okay, you can share your screen.
Can you just check this one, please? You have to do it in the PowerShell or in the Ubuntu?
Ubuntu.
Okay. So I tried this one and it is coming in this. This says the following packages are not available from current channels. How do I change this one?
Okay. So the problem here is you have not set up the path and what default channel you are going to use. So I'm going to work with you. Okay. I'm going to paste this in the chat. So what you
Okay.
need to do. Let me copy the code. So I'm going to paste in the chat. I'm writing code.
Okay. We just copy paste, right?
Yeah, then I will paste in the chat and then you can copy and paste.
Yeah, okay.
So first, what you need to do is activate your Conda environment.
Okay.
Okay, activate your Conda environment that you have created yesterday. So I'm pasting here, see in chat. So you can replace S&P analysis with your own name. What was the name of your environment? See in chat. Copy this command and right click.
Honda activate SPN analysis.
Yes, as for SPN analysis, it's up my environment name. So what?
Okay, I will put my
Yeah,
environment.
you need to change it.
Okay. You will paste with the Control C?
No,
Control P?
just right click, copy and then right click.
It doesn't come. Okay, I got it.
Yeah, so
So I will
no
change this.
backspace. Yes, just with the backspace. Yes, write your environment name.
Underscore. I will enter.
Yes.
Oh, that is.
Okay, so you don't know your environment exists or not. So what you can do, type here. I'm typing here and another command in the chat. Copy this and run this command. So it will let you know either you have created your Conda environment or not. Okay, your Conda environment name is project one. No,
Yeah.
yeah, no, we have figured that out. What's your environment name? So run that command again. Conda activate project
Activate.
one. You can copy from chat.
Okay.
Backspace will work.
It's exactly as it is. Project.
You can see below the base, your environment name is project one.
Yeah, project one.
Yes. No, press enter.
Mhm.
No, there's no analysis. You can see there's no
Okay.
analysis.
Okay, okay. It's just that?
Yes, your project, you can see your project one is environment name.
Yeah, okay. Project
S&P
one I got.
analysis, it was my
Okay, okay.
my
Oh, I'm so sorry.
environment
Okay.
name.
I got it. Okay.
Okay. Okay.
Oh, that's a
Good.
mistake I did. Okay, okay.
Yeah, no problem. So can you now
Yeah, we
type
got it.
Conda install fast QC? Let's see.
Yes.
Okay. No, we need to set the channels. So what that channels means, it will connect to some web page where it will download the QC and then install it. So
Mm-hmm.
first we're gonna set that. Okay, don't worry. Okay, so run this command in the. I just pasted in the chat.
I will just run
I
the.
have a question. I'm having problem with environment setup.
Okay, okay. After I'm helping her, then I can come to you.
Okay.
Okay.
Add channels by Conda. I will press enter.
Yes, and then there is another command I have pasted.
Okay. Yeah.
Okay. No, try again. Conda install fast QC.
Okay. Conda install.
Yes, I'm going to paste in the chat.
Okay.
It's in the chat.
Yeah.
Okay, it's
Yeah.
working.
Yeah.
Can
It's
you
going.
please
Yeah. It's going.
yeah, can you please no stop sharing? Then I can help
Sure.
other one.
Yeah. Thank you so much.
Yes,
No problem.
please. I'm having problem.
Sorry, what's
This
your
is
name?
my Dr. Sadia Ijaz.
Uh, okay, Dr.
And
Sadia.
okay.
Don't worry, we can figure that out. Don't worry.
Uh, this is my screen,
Okay,
and I'm having problem with setting up my environment. It's not recognizing, although I have this.
okay. Let me see.
Okay.
Can you please run this command? So we need to first figure out is there any environment exist or not. I have just pasted one in the
Wait,
chat.
wait. Okay, one second. This command.
I just can you see in chat?
Yes, this command.
Yes, this one.
Okay, it's giving me this.
Okay, you don't have any environment. You can see you have not created any environment.
Okay, so how
Okay.
am I going to do that?
Yeah, no, I'm gonna help you to create environment.
Okay,
I'm writing a code and pasting in chat.
okay.
Okay, Dr. Sadia, here's the code,
Yes.
and after name, you can type any name you want to write.
Okay.
Can you see in chat?
Yes, yes.
Yeah. After name, then you will write, give space. No. Just name. Name should be as it is. No. Don't delete the name.
Name?
Yeah, don't delete the
Any
name.
name? Any name?
So
Any
name
name?
is the part of command. After name, you will give
Okay.
space and then you will, okay. There's a no
Okay,
space
okay.
between hyphen and name.
Okay.
Yes,
All
that's
right.
that's correct. That no, you can write any name.
Okay.
Yes, press enter.
Okay. Then okay.
Okay, type VY. Great. Okay, now your environment has created. No, you should activate your environment.
Okay.
Conda activate. No. Conda activate. And your environment name.
Okay.
Okay.
Is it okay?
Yeah. No. What? Okay.
Your new environment
Type
name
this
is
command.
Genome.
Uh, you can see in chat, type this command.
Okay.
So you can see you just created your environment and you have given the name genome, not the project
Okay.
one.
All right. All right.
Yeah.
So I have to
Hello?
write
Yeah. Conda.
Conda activate genome.
Yes, genome, the environment name.
Okay. Okay. Okay.
Hello, this is Mahrukh. I have same problem. It says command not found when I am creating my environment.
Okay.
Okay, no
All
problem.
right. I get it now. I
Okay.
get it now.
Okay. And, Dr. Sadia, can you proceed to installation of FastQC? Then I can help other
All right.
colleagues.
Okay. Thank you so much.
No problem. So can you please start sharing your screen so I can help you?
Sure. Can you see my screen?
Sorry, what's your name?
Mahrukh.
Okay. No, I'm not able to see your screen.
Okay. Wait a minute.
It's
Now, can you
it's
see it?
Yeah, I can see now.
excuse me. Excuse
Okay.
me.
Yes.
It's still saying no packages found error.
We need to set the path of your Conda path. So can you
Can you
run
please
these two
share
commands
the?
I'm pasting in the chat?
Oh yeah. Okay.
So we need to add the channels.
Okay.
Okay. So, okay. Mahrukh, can you type this command? So we should first see, okay, is there any environment
Like,
exists or not?
okay.
Hello there. I also could not activate Conda. Please help me out.
Yeah, sure. I can help you.
It's still saying command not found.
Okay, can you please type Conda version like this? I'm typing in chat.
Okay.
And can you maximize your screen?
Sure.
Yeah, no, I can see everything. Okay, so it looks like you have not installed Conda.
Okay.
Yeah, so what you need to do is just close it
Okay.
and go to your Windows search. Just click on Windows search.
Okay. Then
Okay, then type PowerShell. Okay, just wait a second. You have already installed Ubuntu. Just open Ubuntu. No, open Ubuntu.
can you see it?
I'm not able to see your screen.
Now can you see it?
It's coming up.
Okay.
Okay, now I can see your screen. So first, what we are going to do, we're gonna install Conda. Okay, so I'm
Okay.
writing code for installation of Conda. Okay, and then I'll paste in
It's the
chat.
same like we get command, right? I
Yeah.
did it, I think.
Yes, mini Conda is there, but we need to install it. It means you have
Oh,
downloaded, but you have not installed it.
okay.
Okay.
Okay. It's the same command like we get. I have that command. So I can
Yes,
paste it. Okay.
you got it, but you need to install it.
Oh,
Excuse me, sir.
Yes.
Sir, she has written ls. What does ls means?
Yeah, ls means list. It means the folder you are in, it will enlist everything.
Okay, sir.
Okay, first run this command and then I'm writing other one.
okay.
Okay, these are the two commands you need to run. You need to run one by one.
Okay.
बाद में खा लियो
Now can I run the other one?
Yes, sure. Oh, you have copied everything. Yeah. No, you can. Okay. No, you can. Yeah, copy the second one.
Okay.
So press enter.
I did the same thing yesterday, but I don't know why it's not creating my environment.
Okay, no problem. We're gonna figure it out. Don't worry.
Okay.
Okay. Keep pressing
Okay.
more.
I think I should press yes or no.
Just yeah, okay. Press yes. Okay. Press enter.
Enter.
Yeah.
Now should I create my environment?
Can you please maximize your file?
Can you see it?
Yeah, there's a pop-up, this stop sharing pop-up. Can you relocate it to any other place? Yeah, or hide it.
Okay. Now can you see it?
Yes, no, no, I can see it. Okay. Can you type again conda dash dash version? Okay, just type conda.
Enter.
Yeah. Okay, I think okay, let me write another code.
Okay.
Okay. Can you we need to first fix the path. Can you type this command?
Okay.
Okay. Can you type this again?
The same.
I have pasted in conda info
Okay.
environment. So I don't know what's going on, but your Conda is not installed. So we can try one more time. Okay. Then what we gonna do?
Okay,
I can, yeah, we can do a personal Zoom session. Then I will help you to install.
sure. Thank you.
Yeah. So can you try one more time installing Conda? Just
Okay,
download it. Try to download it again. So here's the wget command.
yeah.
Okay. No, run these two commands. Okay, press enter.
Yeah, I'm doing that.
Okay.
Now the Conda version.
It has not installed. It has not installed.
Yeah, there is an error.
So can you
Sorry, I cannot hear you.
I'm not speaking. I'm just reading what it says. Okay. So there's a problem with the path. So we can communicate separately. Then I can fix it.
Okay, thank you.
Okay. No problem. So there's a path error. It's not a big error. We need to fix the path.
Okay.
Okay. Okay.
Can I share my screen so that my problem can be fixed?
Sure.
It says I'm trying to activate my environment, but it cannot. Please help me out.
So what you are trying to do?
On the activate my environment name, and
Okay.
it appears error with unrecognized argument.
Okay, let me see.
You're already in your environment.
So you have, I think, yes, you have already installed, you are in environment, but if you want to confirm, type this command. I've just pasted in the chat. So you have created EN77 environment. You are already in your environment. So you know you can install multi QC or fast QC.
I have to paste this phone line for ENVS.
Yeah, you can paste here and see how many environment you have created. So you can see you have created one environment EN77 and you have already activated your environment.
Now,
So
what is the next step?
so you need to install fast QC.
Fast QC, and what is the command for it?
Let me type again in the chat. Here it is. Before that, if it create any problems, then you need to add the channels. First, run these two commands.
Add channels.
Yes.
So to search for the alternate channels. Okay. One by one, I have to run these commands.
Yes. Yes. To add channels to your Conda environment, so it can recognize and retrieve from internet. Okay. So type this.
Now what's next?
So it's installing. So you can see it's working. Okay. I think now we can move forward to the next step of our pipeline.
I have to type Y for yes.
Yes. Okay. Okay, you can see the next package we are going to install is multi QC. So simply what we are going to type conda install multi QC. So this one. I'm going to type in
On the
chat. Okay. Next package we are going to install is trim glare. So trim glare, it is used to trim filter or remove the adapters in our reads. So far, anyone has any question?
please look at my screen. There is some problem.
Okay, you can share your screen.
I am sharing my screen.
Wait a second, I'm replying in the chat. Okay, now I can see your screen.
It says right here, trim galore is not installable because it requires cut adapt, but there are no viable options.
Okay, the command I have shared, you have tried to install it. Can you maximize your window?
No. It is maximized.
Okay. Type this command to install trim glare.
Which command? In the chat?
Yeah.
No recent command. If you're looking at my screen,
It's in the chat.
which one? This one? This
Yes.
one?
Yes.
No such file or directory.
Okay. Type conda install. Let me write a code. Try this channel, buy conda channel. I've pasted in the link.
I haven't received in chat.
Yeah, it's in the chat. Sorry.
It's not.
Oh,
Okay, could not solve for environment specs.
okay. Wait a second. Okay. Um, it is because of some Windows issue. Let me write another code for you. Okay, let's try updating your conda. Can you update your conda?
Excuse me, sir. There's
package is not installed in prefix.
Okay. Okay, let me write another code.
So I'm having the same problem with the trim glare. Can I do the same coding?
Okay. Which windows you have?
I'm sorry, I don't know. It's Windows 11.
Okay, okay. Let me see.
I think it's taking a lot of time.
Okay.
If you don't mind, we can have a separate session so that my problem can be
Okay,
resolved.
okay. So, okay, we can move forward.
Yes.
Okay, so after installation of trim glare, okay, there's another package that we can also install is Trimatic. So for Trimatic, the drawback with the Trimatic is you need separate adapter file. So trim glare has all inbuilt adapter file. You don't need to provide it separately. So these are the two packages that we can use for trimming and filtration of our reads. Okay. So next thing, what we do. So we have option. We can remove unwanted RNA like sort me RNA. It's another package that we can install and use it for removing unwanted RNAs. Okay. So next thing what we do is install star. Star is used for alignment. So we can install the package star, and then using that star, we can do the alignment. So after doing alignment with our reference genome, we first we have to index the environment. In indexing mean then it will be ready to align with our reads. After alignment, then what we will do, we will get a SAM file. So SAM file, it will be converted into BAM file, a binary file with system can read and process easily. So after getting BAM file, then we need to sort the BAM file. After sorting, what we will do, we will use the module called or package called subread. So this subread has a feature called feature counts. So using feature count and using GTF file. So GTF file is annotation file. So it is really important to provide the GTF file, I mean annotation file. So once you have provided annotation file, what you will get a text file which will contains your read counts. So no, I'm going to show how a read count file looks like. Let me share another file. So this is the file you will get at the end. So you can see these are the gene IDs and these are the reads. These are your samples. It actually it's a one sample and there's other replicates. So you will get the reads. So next step, what we have to do after getting the reads, we will process these files in R Studio or using R. Either you can use the R in Linux or you can use the R separately. So now I'm going to show the R script. So R is another programming language. You can see this. This is the R Studio. So what we are going to do use in this R Studio, there are two packages we use, app.glm and DESIC. So the reads file we get from after doing all running all those Linux commands. So we'll get we will run DSIG, and this one is a script. So I will share the script. So you will run the script, and at the end, what you will get differential expression. So how differential expression data looks like? Let me show. I opened too many files. Let me see. What's that? Okay, this is what we got after doing differential expression in R. So you can see we will get base, mean, fold, change, and other quality parameters. So these are the all gene annotations, so which we have provided with our GTF file. Okay, so after getting this differential expression file, then we can do further analysis and visualizations. So now what I want you to do, so download your any FASTQ files from NCBI and then try to run. So let me share that pipeline with you. So I'm going to type all packages we need to install in using our Conda. So you can see what packages we need. FastQ we have already installed. Then for trimming and filtering of read, we need Triomatic, or the other we can use is TrimGlower. So for sorting unwanted RNA, we need to install SortMe RNA. And for indexing and aligning, we need to install STAR. And for processing our files, like if we want to convert SAM to BAM, or other want to count the reads, so we need SAM tools. And then other last one, we need subread. It's a package called subread. So I'm going to paste all these in chat. So number one, we need FastQC. Number two, we need multi-QC. Number five, we need STAR. Number six, we need and number seven, we need so maybe there's a Windows version problem or some R environment problem. So other we can install is the other one for sorting our sequences. So I'm going to write code for TrimGlair. So you can, Kiran, you can try installing the other package, Trymatic. So I have already pasted in the chat. So anyone please can share the screen who is trying to install.
Sir, I have the same issue that Mushra has. You
So
have said that it's
what you are trying to install?
sir a trim glare.
Okay, let me see if I can get the control. Okay, let me write another code.
Okay, sir.
Okay. Try this code. So you're still getting the same error. Okay.
Yes,
Try
sir.
to try to create new environment with Python.
So can you share the command?
Yeah, I have just typed in the chat. Can you please try this command? Conda create with Python 3 point with Python. We type y. Okay, try to activate. I hope it will work now. Activate your new environment. Conda activate my env. Conda activate my env space. Okay, now try to install Python. Conda install Python. No, sorry, Python is already there. Now try to type these commands. I have pasted in chat.
Okay, sir.
Yeah.
Do I put the commands one by one or
Yeah,
all
just
at once?
copy and paste. It will detect if you need to do one by one or together. No, copy in the its command is in chat. Copy and right click. Yeah.
The same happened with me while installing Python. Just it is visible on the screen of.
Can you check your RAM? What the system requirement is? Your RAM should be at least 4 GB.
Okay, let me check.
Okay, you have 32 gigs. That's wonderful. Okay, there's no problem with storage or any with any other specification.
I
So
have 8 GBs of RAM in my system, but still it's not working.
okay, so what you can do? Can you copy all this error and paste in the chat? Send it to me in chat. Copy that all error you're getting. So I'm not able to read. So paste in the chat. Let me read it. Yes, Control C. Yeah, copy with Control C. Hmm.
I tried copying with Control C, even then it's not being copied.
Just select all, then press type Control C, and then in chat Control V.
So it's giving error that your message is too long.
Same here.
Okay, okay. Let me. You can paste two three lines. Yes, yes. Paste all. Okay, could not solve environmental specs. Following packages are incompatible. Pin 1 install and its Python 3.3 can be installed. Trimgler is not installed because it's required. Okay, paste all line. Paste one by one all lines. Okay, the next thing we require is cut adopt Python 2.7, Python 3.4, cut adopt 1.16. Okay, Python Python 3.8 is conflicting with other. Okay, okay. No, I can fix it. Let me write another code.
Okay, sir.
Okay, so first what we are going to do, we're going to create new environment. So I have just pasted in chat. Okay, type yes. Okay, now activate the new environment. type the second code, conda activate en88. Type en88.
Sorry, sir, again, can you?
en88. Okay. Now type other command I have pasted in chat. So we are going to see the first problem we are going to resolve is Python. So we have created environment with Python. No, the error code you have pasted, it required cut adapt. So now we are going to install cut adapt. Okay, type yes. Now type this code. Okay. Now we will be able to install. Now install again. Okay, now it's installed.
Okay, sir.
Yeah. So next thing, so all you need to install all other packages to run this pipeline one by one. Multi-QC, star, SAM tools, and subreads.
So can you share the command or the how to write the command for
Okay.
multi-CQ and
Okay. So type conda install star. Oh, you misspelled the conda. Okay. No, you're fine. You can install everything. Okay. Next, install SAM tools. Conda install SAM tools. Okay. Okay. Next, install subread.
so the spellings are right?
Yes.
subreads.
Yeah. Okay. Conda install subread. Do not write, just subread, not reads, just subread. Yes. Okay. Yes. Okay. Now you have everything that we need to do our analysis.
Okay, thank you, sir.
Yeah. So it's for everyone. So these are all packages we have learned to install. So next thing, what we need is data set and do analysis. So I'm gonna tell you the what are the minimum resources we need to do all these analysis. At least minimum you should have 1 TB storage and 16 GB RAM, and your processor should be, if you are using Windows, it should be above i7. So if you have these kind of resources, you can easily perform all your analysis on your computer. So if anyone has any question, so next up, what I'm going to do is share the pipeline, how we can run the pipeline. So you can download any data set and then follow the pipeline, and you can do the analysis by your own. So far, if anyone has question, feel free to ask.
Sir, can you please tell us about how we can download the RNA-seq file from the NCBI?
Okay.
FASTQ.
Yes. So what we okay. So if you go to NCBI SRA, you will find all RNA-seq data. So you can see I have just typed Arabidopsis, and you can see 18 some around millions of data sets you can get from NCBI SRA. So the good thing about SRA, you will get raw reads. So you can see these are the platform. For example, if I'm clicking Illumina and RNA-seq data, so you can retrieve RNA-seq data, and then what you can do your analysis. So any other question? So I've just shared the pipeline in chat.
So we can't see any.
Let me see. Oh, it went this direct message. Okay. So if anyone has any question, we can address that question. Otherwise, we're gonna close this session, and then next we will in our last session we will do de novo assembly and variant call.
Please share the commands for you mentioned.
Yes, I have
Star
shared
and
in chat RNA-seq pipeline.
pipeline. Okay,
Yeah.
thank you.
So Saleh has asked a question. You mentioned that system specification, terabyte storage are something. That's what we needed to further process the RNA-seq analysis. So the RNA-seq data is always in GBs. So you need a good amount of storage. Like it should be around minimum 1 TB, 2 TB storage. So your system can handle, and you require a RAM around 16, 32 GBs or 64 GB. Depends on how faster you can do. If you have a minimum RAM like 16 GB, you can still manage, but it will be really slow. It will take days. But you have higher RAM like 32, 64 GBs, then you can do it easily. Okay, everyone. So we are going to close this session. Thank you so much for being the part of this session. Okay, bye everyone.
Thanks to you, sir. Allah Hafiz.
Bye.